Cell. All living things are composed of cells, and a single cell is the smallest unit that has all the characteristics of life. It is the structural and functional unit of the living body. Although cells in the body differ in size and shape, they are basically composed of the following components: cell membrane, cytoplasm and organelles, nucleus with chromosomes. Cell membrane. The cell membrane or plasma membrane is a thin layer of biological membrane that separates or protects the cell from the outside environment. It is principally composed of lipids, 40%, proteins, 55%, and carbohydrates, 5%. The basic structure of the cell membrane looks like proteins floating in a sea of lipids, commonly referred to as the fluid mosaic model. Lipid bilayer. The whole portion of the membrane is composed of bilaminar of lipids. These lipid molecules consist of phospholipids, cholesterol, and glycolipids. The phospholipid molecules consist of two regions and resemble a headed pin. The globular end of the molecule is called the head, while the other part is termed as its tail portion. The head portion is soluble in water and hence known as the hydrophilic or polar region. The tail portion is insoluble in water and hence known as the hydrophobic or the non-polar region. These hydrophobic tails remain enclosed within the bilayer, while the hydrophilic head extends outside on either side of the membrane. This lipid bilayer structure acts as a barrier against the majority of the water-soluble molecules. They are, however, selectively permeable to fat-soluble molecules such as oxygen, fatty acids, and alcohol. Cholesterol molecules are located in between the phospholipid molecules. The cholesterol molecules support the package of soft and oily phospholipid structures within the membrane. The structural integrity of the lipid bilayer membrane is maintained by these cholesterol molecules. Proteins. The cell membrane protein constituents float within the lipid bilayer. Most of these proteins are glycoproteins. Based on their arrangement, protein molecules are classified into two categories: integral proteins or transmembrane proteins, peripheral proteins or peripheral membrane proteins. Integral proteins. Integral or transmembrane proteins are the proteins that pass through the entire thickness of the cell membrane from one side to the other side. These proteins are tightly embedded within the bilayer of the cell membrane. Peripheral proteins. The proteins present on the outer and inner surfaces without penetrating into the bilayer of the cell membrane is known as peripheral proteins. They are loosely bound either to the integral proteins or with the lipid layer. So these protein molecules dissociate readily from the cell membrane. Peripheral proteins may be intracellular peripheral proteins, which are located on the inner surface of the cell membrane. Extracellular peripheral proteins are situated on the outer surface of the cell membrane. Carbohydrates. The carbohydrate molecules attached to the cell membrane proteins are known as glycoproteins. Those carbohydrate molecules attached to the lipids are termed as glycolipids. The entire surface of the cell membrane is covered through a thin and loose covering of carbohydrate molecules which is known as glycocalyx. The main function of these carbohydrates are to facilitate cell signaling or interaction and also provide protection for the cell membrane. Functions of cell membrane. The major functions of the cell membrane are to act as physical barrier and to regulate the exchange of substances from its surrounding environment. Cytoplasm. The cytoplasm of the cell is the jelly-like material formed by 80% of water. It contains a clear liquid portion called cytosol and various particles of different shape and size. These particles are proteins, carbohydrates, lipids, or electrolytes in nature. Cytoplasm also contains many organelles with distinct structure and function. Endoplasmic reticulum. The endoplasmic reticulum is a eukaryotic organelle with an interconnected network of flattened membrane-enclosed sacs or tube-like structures called cisternae. The fluid that fills the interior spaces is known as endoplasmic matrix. The endoplasmic reticulum is of two morphological types, rough and smooth. The rough endoplasmic reticulum's outer surface is covered with ribosomes and acts as the site for protein synthesis. Hence, they can be prominently found in cells such as hepatocytes. The other type of endoplasmic reticulum is smooth. It lacks ribosomes and functions in lipid manufacture and metabolism, the production of steroid hormones, and detoxification. They can be found in large quantity in mammalian liver and gonad cells. Golgi apparatus. The Golgi body or Golgi complex is a membrane-bound organelle involved in the processing of proteins. Each cell has at least one Golgi apparatus and is situated near the nucleus. Each Golgi apparatus consists of 5 to 8 flattened membranous sacs called the cisternae. It has two ends or faces, namely cis face and trans face. The cis face is positioned close to the endoplasmic reticulum. Reticular vesicles from endoplasmic reticulum enter the Golgi apparatus through this face. The trans face is located near the cell membrane. The processed substances are transported outside from the Golgi apparatus through the trans face. Functions of Golgi apparatus. They serve as the wrapping and packaging department of the cell. They are the site for lysosome synthesis. They facilitate carbohydrate addition to proteins to form glycoproteins. They produce secretion granules for storage of hormones and enzymes. Mitochondria. The mitochondria is a eukaryotic organelle found in the majority of the organisms and have a double outer membrane. Mitochondria have been described as the powerhouse of the cell because they synthesize ATP, which is the source of energy. Mitochondria are globular in shape and are bound by a double membrane, the outer membrane and the inner membrane. The outer membrane is smooth and encloses the contents of the mitochondria. It contains respiratory enzymes for biological oxidation. The inner membrane is folded to form cristae, which project into the interior of the organelle. And this contains enzymes related to the metabolism of ATP. Mitochondria has its own DNA that can undergo autonomous replication. Functions of mitochondria. They utilize ATP and generate the energy required for cellular functions. Ribosomes. Ribosomes are small granular structures which are composed of 35% proteins and 65% ribonucleic acid, which is termed as ribosomal RNA or rRNA. The ribosomal proteins and rRNAs consist of two separate fragments with different size. These are known as large and small subunits. There are two types of ribosomes. Ribosomes which are attached to the endoplasmic reticulum, ribosomes that are freely found in the cytoplasm. Functions of ribosomes. They help in protein synthesis. Endoplasmic reticulum-bound ribosomes synthesize all the transmembrane proteins, secretory proteins, and proteins stored in Golgi apparatus, lysosomes, and endosomes. Free ribosomes synthesize cytoplasmic proteins such as hemoglobin and proteins found in peroxisomes and mitochondria. Lysosomes. Lysosomes are the membrane-bound vesicular organelles found throughout the cytoplasm. The lysosomes are formed by Golgi apparatus. The enzymes synthesized in the rough endoplasmic reticulum are processed and packed in the form of small vesicles in the Golgi apparatus. Then, these vesicles are pinched off from the Golgi apparatus and become the lysosomes. It has many small granules which contain hydrolytic enzymes. There are two types of lysosomes: primary lysosomes and secondary lysosomes. The primary lysosomes are vesicles formed by Golgi apparatus and are enclosed by a single phospholipid layer. They contain acid hydrolases, which are important in activation or deactivation of the enzymes. Most of these fuse with phagosomes and develop into the secondary lysosomes. These secondary lysosomes are larger in size and convert the inactive enzymes into its active form in order to release them outside the cell. The enzymes are grouped based on their function into nucleases, proteases, glucosidases, phosphatases, lipases, and sulfates. Cytoskeletons. All cells contain a cytoskeleton, which is a complex network of interlinking filaments and tubules that extend throughout the cytoplasm from the nucleus till the plasma membrane. The cytoskeleton is a dynamic structure composed of three main proteins: microtubule, intermediate filaments, and microfilaments. Microtubules. Microtubules are straight, hollow, and long tubular structures formed by globular proteins known as tubulins. They consist of alpha and beta tubulin polymers. These subunits aggregate to form long tubes made of stacked rings. The transportation of vesicles and organelles within the cells occurs through the formation of tracts. The microtubules also assist in chromosome migration during mitosis by spindle formation. The molecular motors assist in the movement of proteins, organelles, and other cellular contents. The molecular proteins, dynein and kinesin, utilize microtubules for the cytoskeleton movement. The interaction of these motor proteins with tubulin mediates the movement. Microfilaments. Microfilaments are long filaments with 6 to 8 nanometer diameter made of actin proteins. They attach to various parts of the cytoskeleton and interact with the membrane-bound proteins. They also assist in the movement of myosin molecules. Myosin attaches and walks along the microfilaments. The myosin motoring along the filaments, called F-actin, generates contractile forces in the so-called actomyosin fibers. Intermediate filaments. Intermediate filaments are filamentous structures around 10 nanometers in diameter. These filaments are considered to be more stable than the actin filaments and are also the heterogeneous constituents of the cytoskeleton. Intermediate filaments are responsible for the structural organization of the cell. They anchor the organelles and also serve as the structural components of the nuclear lamina. The absence of intermediate filaments results in easy rupture of the cells. Molecular motors. The molecular motors are motor proteins involved in movement of different organelles, proteins to other parts of the cell. They are ATPases of 100 to 500 kilodalton, and its hydrolysis causes the molecules to move. They are classified into two types based on their association with the cytoskeletons: those producing motion along the microtubules and those around the actin. The microtubule motor proteins are in turn divided into two types: kinesins and dyneins. The kinesin proteins carry the membrane-enclosed organelles through axon terminal, walking towards the plus end of microtubules. The dyneins are minus-end directed microtubule proteins which are further categorized into two groups: cytoplasmic dyneins and axonal dyneins. The cytoplasmic dyneins facilitate the vesicle trafficking and Golgi apparatus localization near the center of the cell. Axonal dyneins carry out the sliding movement of microtubules and thereby assist in cilia and flagellar beating. Nucleus. The nucleus is the largest membrane-enclosed organelle found in eukaryotic cells. Eukaryotes frequently have a single nucleus. However, a few cells such as mammalian red blood cells don't have a nuclei, whereas others have many. Generally, the nucleus is spherical in shape and located in the center of the cell. Nuclear membrane. The nuclear membrane consists of an inner and outer membrane arranged parallel to one another, resembling the bilipid layer of the cell membrane with interspersed proteins. The nuclear envelope protects the cell's genetic material and separates it from the surrounding cytoplasm by acting as a barrier. This also prevents the diffusion of free macromolecules between the nucleoplasm and the cytoplasm. The pores on the nuclear membrane are composed of multiple proteins and are collectively referred to as nucleoporins. Nucleoplasm. Nucleoplasm is an aqueous substance present in the nucleus and is similar to the cytoplasm of the cell. The nucleoplasm is enveloped by the nuclear membrane and contains chromosomes and nucleoli. Many substances such as nucleotides that are necessary for the replication of DNA and the necessary enzymes for these activities are dissolved in the nucleoplasm. Nucleolus. The nucleolus is a non-membrane bound suborganelle present within the nucleus. The nucleolus is formed around tandem repeats of rDNA and DNA that codes for the ribosomal RNA, rRNA. Their main function is to synthesize rRNA and assemble the ribosomes. Chromatin. The chromatin consists of a complex of macromolecules such as DNA, protein, and RNA. Histones are primary protein components of the chromatin that compacts the DNA. DNA wraps around histone proteins, forming nucleosomes. The beads on a string, commonly called euchromatin. The multiple histones molecule wraps into a 30 nanometer fiber consisting of nucleosome arrays, which form the most compact structure called heterochromatin. This can be called as the chromatin fiber. The chromatin fiber further condenses to form the chromosome. DNA. Deoxyribonucleic acid is a molecule that carries the genetic information necessary for the growth, functioning, development, and reproduction of all known living things. They are made up of nucleotide molecules, which consists of a phosphate group, a sugar group, and one of the 4 types of nitrogen bases. The phosphate and sugar groups form alternating chains that form the strand of DNA. The 2 strands are interconnected through nitrogen bases found in nucleotides, which are adenine, thymine, guanine, and cytosine. The arrangement of these nitrogenous bases determines the biological instructions encoded within a strand of DNA. The complete set of DNA molecules in humans contains about 3 billion bases that are dispersed as a set of about 20,000 genes on 23 pairs of chromosomes. The 2 alternative strands of DNA run in anti-parallel directions to one another. The DNA molecule forms a double helix structure, which resembles a spiral staircase. This structure arises as a result of its secondary structure. The double helix consists of about 10 to 10.5 nitrogenous bases pairs per ton. Chromosomes. Within eukaryotic cells, DNA is organized into long structures called chromosomes. During cell division, these chromosomes are duplicated in the process of DNA replication, providing each cell its own complete set of chromosomes. RNA. Ribonucleic acid, RNA, is a polymeric molecule that is essential in several important biological functions. Similar to DNA, The RNA molecule is also assembled as a chain of nucleotides. However, unlike the DNA, they are frequently found as a single-strand molecule in nature. In RNA, the nitrogenous bases are guanine, uracil, adenine, and cytosine. Thymine of DNA is replaced by uracil. Several viral genetic materials are encoded by an RNA molecule. The RNA molecules play several significant roles, such as catalysis of biological reactions, control and regulation of gene expression, sensing and communication of appropriate responses to cellular signals. Among these, the protein synthesis occupies as the most important function of RNA molecules universally. There are three types of RNA: messenger RNA, mRNA, ribosomal RNA, rRNA, and transfer RNA, that is tRNA. Among these, rRNA constitutes around 80%, tRNA about 15%, and mRNA is nearly 5%. The mRNA carries the genetic information to be coded into proteins. rRNA forms the ribosomes, which is the site for protein synthesis, and tRNA function in transport of amino acid during protein synthesis.